Can everybody see my screen okay here?
Yep, that looks great.
Okay. All right. Yeah, so the title says it all, packages in progress. And so I have had the idea to try to come up with an R package for automated detection for quite some time. And so that's kind of where I am. And I guess the big kind of push as to why I originally wanted to do this is that I wanted to understand how the detectors and classifiers were working. And let me see if I can move ahead here. So I originally developed a package called gibbonR. And so those of you that don't know me, I'm a gibbon researcher. They're small apes that live in Southeast Asia, and they sing these really loud, long, beautiful songs. And so that's kind of where the name came from. It's what I study. And so I originally developed this package to have, and I realized later that it kind of had too much all at once. And so I initially developed this package to automatically detect and classify acoustic signals, and then also to do some data visualization. So basically creating biplots to show differences in acoustic signals. And so I have since decided to split them up into two. And so the second one, which I have called now, and I'm also up for recommendations for better names if you guys have them. The second one is gibbon ID. And so that one is going to be focusing on the data visualization part. And so since I developed this detector classifier in R a couple years ago now, there have been some papers published doing what I did and better. So, I'm kind of at the point where I'm still excited about trying to move these projects, especially the detectors and classifiers, forward. But I need to be careful, and I also am hoping to get some input from all of you about how I could make this something that is useful. And I guess the one thing I didn't mention is that, and I'll talk about how the detector and classifier I developed works, but it doesn't rely on deep learning models. So it's not really like a fancy, sophisticated one, but I'm hoping that there's still potential for it to be useful. And if I can think about how to kind of frame that, then I think that would be good. And I would love to get your input. And so I kind of mentioned already, so my goals for today, I'd really love to get from the community your ideas on kind of what types of updates I could maybe bring about, and the types of functions that maybe you would like to see. And then, yeah, I mentioned this already. So in light of some of these new applications, how can a more simplistic kind of detector / classifier still be useful? And I think it can be, but I need to get some thoughts from you all. And then, so the gibbonR, it's on GitHub, and there's a preprint out, but it's undergoing pretty major updates. So I think if you're interested, please wait to download it, and you can send me an email, and I can let you know once the updates are available. And so it was interesting. I was like still learning how to use GitHub when I was first developing this package. And so I was like pushing all of the changes, and then I was getting a lot of interest just from the what I had on GitHub. And so that was the kind of major push for me. I was like, okay, I should probably get a preprint out on this. And I think this was before COVID, when everybody knew what preprints, what they were and the potential impact that they could have, and how widespread the information can become. And so I think if I would have known how broadly and widely kind of viewed it would have been, I would have maybe waited a little bit to put it up on as a preprint. So that was kind of a learning experience for me as well. So that was, just a few years ago, and we're all learning in this process. But anyways, so it's undergoing major updates. So please, if you're interested, you can send me an email, and I can put you on the waiting list for when it's going to be available. And So I was hoping before we delve into some discussions on the specifics of the package, if people could unmute and tell us, or if they could potentially put in the chat what kinds of automated detectors or classifiers they have used, and what you liked or did not like, and if there are any functionalities that you wish they had that they didn't. And I can't see everybody, so you don't necessarily need to raise your hands, but can I just unmute?
Hi, Dena. This is Vijay here.
Hi.
Hi. I think I might have emailed you about your package a while ago. Oh, and one of the things I think, especially after using Maslow's package as well, which I would really love is parallelization, especially of some of the detectors. And if there's an option to, parallelize the processes across multiple cores. Yeah, and yeah, I think that's definitely one functionality that I would love to run, given, I mean, if we have a lot of data, especially.
Yes, thank you. No, that's a fantastic point. And I have, with the current version I've been playing around with kind of different ways to try to speed up the analysis. And yeah, the idea of trying to get it on multiple cores is definitely one that I have been thinking about, that I haven't quite gotten there yet. One thing that I did, and this is maybe specific to kind of how my detector works, but as opposed to like my recordings were like 1 to 2 hours long. And so what I would do is like cut them into shorter time chunks, and then it could analyze those a lot quicker than like reading in this big sound file. So that was one way. But yeah, the idea of the parallel cores and trying to get it faster, that's a great one. And I don't know about like I think Connor said he couldn't be here, like the BirdNet team. I'm kind of curious how they are dealing with that, dealing with the data processing part. Like I think they're using maybe high performance computing, but yeah, it's something that I think we're all kind of dealing with. So anyways, thank you for that suggestion. I see Kathleen says she loves the simplicity of template-based detectors, but they're not great for all sound types, especially if there's a lot of time frequency variation in vocalizations from a target species. Yes, Kathleen, thank you. And so I know Raven has a template detector. Has anybody used the ones in R? And I know Kathleen, you have, right?
Yes, I definitely have. Yeah, and I think that about sums it up. I think I love the low barrier to entry, and it's great if you have a really typified call type. But if you don't, it's not really the solution I would look for.
Yep.
But yeah, they definitely have their place.
Yes, definitely. And so my understanding with the template detectors and the ones that I have used, and Russ, I know that you're here and you've done a lot of work with the template detector in Raven, is that you give it one or a couple signals and it does something similar to spectrogram cross correlation, where it kind of tries to find matches based on that. Am I correct in that description?
Yes, exactly.
Yeah. Is there a way, like, so for signals that are more variable, I mean, is there a limit to how many different types of signals you can put in as templates? And then
you can create a template detector in Raven that has many different templates, but the two downsides of, and that can be useful up to a point, depending on how variable the signal is. Of course, it goes a lot slower as you add more templates, because it's doing all those spectrogram correlations for each template separately. And then the other thing is that people sometimes forget is that each individual template will also drag along with it it may drag along with it its own separate set of false detections. So depending on the situation and what else is in the recording, additional templates may help up to a point, but you hit diminishing returns pretty quickly.
One of the Raven options is also to merge templates, but again, that can have, so you can find 6 calls and look for, I think it's essentially the dominant tendency of those, the place where all of those represent energy in the same places. It has some of the same challenges that Russ mentions. And especially if you have calls that are somewhat variable in duration, you might get essentially the average of those in a way that isn't necessarily effective for finding calls. So that actually worked quite well on frogs, but it's not a guaranteed fix in all cases.
Yep. Yeah, definitely.
One quick thing.
Yeah.
One quick thing I would mention about detectors, like, it's kind of goes a layer back to what the question is. And oftentimes, if we're looking for a presence of X in Y dimension, and that dimension could be space and time, and depending on how precise the detector is, we could accept results in different ways. And again, it's a complicated answer or question I'm getting into. But if our goal is to just look at binary presence or yes or no presence of a species in a location over a time period, then the different application of detectors could be like within the tolerable levels. So template 1, if it just captures one signal of a species, and yes, it's not very specific or not very precise, but it gives you a result, then you can kind of live with that performance. So the performance is related to the kind of question that you're asking from the detector.
Yes, definitely. Thank you for bringing that point up. It's always about the research questions, right? And so I see in the chat, Ashik, you mentioned the band limited energy detectors as well. And so my understanding of the BLED detectors is that it scans a certain frequency range and it will pull out anything that's within that frequency range. So it's not discriminating based on anything apart from duration, potentially, right? And then frequency range. And so that can lead, you pull out a lot of signals that maybe are not anything that are related to your target, for sure. So the band limited energy detector on its own, and Russ, you and I have had some chats about this, depending on the soundscape and what's going on, it's probably not going to do great for identifying target signals unless there's only one type of signal in your frequency range of interest, which I guess I have a tropical forest bias. So I don't know, maybe maybe in some other types of soundscapes that might not actually be a big deal, but. Okay.
Yeah, regarding the band limited energy detector, across the universe of all the things, all the types of recordings that people work with in bioacoustics, there's a pretty small set of situations where it works well, because, as you said, it's extremely non-specific. So it basically works well where the background, other than the thing you're looking for, is pretty quiet. For example, it has worked pretty well for some people detecting nocturnal flight calls of birds in temperate regions where the basic soundscape is extremely quiet and you get these little blips periodically. But if there's a lot going on, it's often pretty useless. And Laurel mentioned in the chat an example of doing things like counting pulses of an insect. It can work really great for things like that. But
yes.
it's usually not what we want for most applications.
Yeah, and so I did for my dissertation work for my gibbon calls, that's these long notes that go into these short trill notes. And so I did use the BLED detector to draw boxes around those notes. So yes, there are certain applications where it may be appropriate. Oh, okay, Laurel's saying it's good for finding frog choruses. Okay. Yeah. So I guess, never say never, right? There's always cases when things might work well, but I guess when I'm thinking about the use of automated detectors and classifiers, I'm kind of like thinking about pulling out the calls of a target species from the overall soundscape. But that's also just my bias too. So, okay. Does anybody else have any experience with automated detectors, classifiers? Anybody done anything with the fancy deep learning ones? No, not yet. Connor's not here. Otherwise, I'd put him on the spot and have him tell us about it. Those of you that haven't used the deep learning models, what have been the barriers to entry for that?
I would say in my case, the questions that I've had so far have been specific enough that it hasn't been necessary. But I have recently finally got BirdNet installed after a long odyssey of figuring out how to do it on my particular system. And it's, I mean, obviously that's different from constructing my own deep learning network, but being able to finally apply it and watch it in action has been really, really fun to see it do really well.
Okay. No, yeah, thanks for that input. That's really, it's interesting too. And I think it's a good kind of discussion point. It's like, if the packages or, you know, the approaches you're using are good enough, like, is that okay? And yeah, it totally is. So, oh yeah, and Laurel just put in the chat that Sham is working on a Python package that eventually will be incorporated into Raven to do deep learning for acoustic signals. I see a cryptic code, not very well supported outside of specific projects. Performance is not well understood. Okay, yeah, those are all really great points. And maybe also not just limited to the deep learning models, but maybe some of the other ones as well. Wendy says, getting selection tables together and formatted has been a slog and a stumbling block. Is this is for any sort of detection or for trying to use Sham's deep learning one, Wendy, or?
I mean, specifically related to Shams, but I think it's sort of a universal challenge that probably applies to most platforms.
Yep.
So just figuring out like, and some of it is, you know, starting out with selection tables without having the right columns in them and then having to add those in. And some of it is just like getting everything squished together with the right bit of code.
Yes. No, and I know Marcelo's packages do have the options of reading in Raven selection tables into R and manipulating them that way, right? But yeah, no, I think that's great. Lots of annotated data. Could you expand on that, maybe?
So we've been trying to use this efficient net, neural networks to train our annotated data. But so the thing is, we've been finding it very hard to draw selection, these bounding boxes because it's just a massive chorus, tropical bird calls, etc. We've been using weakly labeled data, and I've been noticing that only the accuracy goes up only to a certain point, and it's actually two things. One is that sometimes even if you have species that have like a wide repertoire, their accuracy seems to be fairly high. But if you have a species like a barbet which calls only in one frequency range, its accuracy sometimes isn't that high. And I think I want to go back to the discussion of, the background probably really makes a difference. I think network is learning something from the background along with what's in the foreground and then sort of predicting higher accuracies because this call or this note is associated with this particular background. I don't know, I'm just speculating.
Sorry, there was a little bit of static. I want to make sure I understood the second part that you were saying. You were a little concerned. Was it about the Raven template detector, that it was incorporating aspects of the background noise as opposed to the signal itself?
No, sorry, I haven't used the Raven template detector. I was just talking about, just in general, I was just using
okay.
Like a
Denver, yeah. Russ, do you have any suggestions or thoughts about how to address that?
Short answer is no, nothing very compelling. I mean, the issue of getting, I'm not an expert by any means in deep learning approaches, but in general, getting any kind of detector, whether it's template based or deep learning, to be focusing on the signal, especially when signal to noise ratio is not high, and the accident of getting things inadvertently overtraining to background noise conditions. I think that's just sort of a universal problem, and I know that people have used various kinds of data pre-processing approaches before you give the data to a network, to train a network, to try to bring out the signals of interest in a sense. But yeah, it's training especially from soundscape, very busy soundscape recordings is always going to be challenging. And of course, the one other thing, I'm not by any means an expert on the innards of BirdNet, but most of you probably know that BirdNet was, at least to begin with, trained, I think, entirely with focal recordings from Macaulay Library and from Xenocanto, where you have extremely high signal to noise ratio recordings recorded with directional microphones. And that worked well, but then sometimes those have trouble with noisier recordings.
Thank you for that, Russ. And I do hope that we can come back to this idea of training data, because it's something that I have been in my applications kind of struggling a little bit with, of like thinking about how to annotate what type of recordings, if we use focal versus PAM, if you're like looking for gibbons or something like that. So anyways, yeah, I think there's a lot of questions around that hopefully I can get a little input. Does anybody have anything they want to add about detectors, classifiers, and their personal experience before we move on? Okay, and thank you all for your input on that. Okay, so now I can go into, and this is a really high level overview of kind of how the detector slash classifier that I developed works. And I guess I want to briefly define or explain my understanding of those two terms. And I know in earlier discussions with Holger, I was kind of confusing and confounding detector and classifier. So how I'm understanding a detector is it's basically kind of identifying signal from noise, and then your classifier is saying this signal is gibbon versus hornbill versus frog. Does that resonate with others, or are there better definitions? I see Russ is shaking his head yes. Okay, I'll go ahead with that. But yeah, it is. I think it's something that is easy to get confused, and sometimes it can be a detector and classifier in one step, or it can be in multiple steps. And I think that's where, when I was first getting acquainted with this stuff, I got a little bit confused. But so the package that I have developed has two steps. So it's first a detector and then a subsequent classifier. And so the first step is we have these long-term recordings. And so mine are from North Borneo. And so the first thing is to basically, within these long recordings, scan the frequency of interest and then say, pull out all of the sound events in this frequency of interest that are over a certain duration. And so I have two different ways that it can do this. The first is the band limited energy detector. And so basically what this does is it creates a spectrogram, and then it calculates for each time window it's the amplitude or the power. And then it will say, clump all of these time windows together, and then we'll say that this is a sound event or not. The second approach is maybe a little bit more analogous to my understanding of how BirdNet works, but it creates a spectrogram just the same, but then for each time window, it uses a machine learning algorithm to say time window, gibbon, not gibbon, or bird, not bird, whatever your signal of interest is. And then it will clump them together that way. So there's two different approaches that we can use to identify sound events. Was that clear? Does anybody have any questions about that? So basically just signal from noise. That's the first step. Oops. And then next, so we have a short sound event. In my case, the gibbon calls are about 6 seconds or longer. And so then we have these sound events, and then we have to convert the sound events into features that we can then feed into machine learning models. And so in this case, I use a set of features known as mel-frequency cepstral coefficients, or MFCCs. They were originally developed for automated speech recognition, and my previous work has shown that they do really good at discriminating amongst gibbon individuals. And so in the next slide, I'm going to talk about all of the decisions that we have to make in this. And, you know, it's funny, I think when I first got into bioacoustics, I was so sure it was going to be just a very objective, you know, science, but we have to make decisions along. And I think that's kind of where I've gotten a little bit stuck in doing the updates. Is like, I don't even know what to set as like useful defaults for all of these different decisions. But anyway, so you have your sound events, and then you extract your features from your sound events. And then you can use your different machine learning algorithms to classify them. So for example, is your sound event a gibbon? Oh, oops, why is this not working? A hornbill, I can't actually see these because of the chat box. There we go. A hornbill, or it probably actually wouldn't be an elephant in this case because they're a lower frequency than, I guess the trumpets actually, I think we might get some of the trumpets, but so that's kind of the overall big picture of how my detector / classifier works. And then lastly, we need human validation of the system. So basically, you know, all right, if it's saying a gibbon, is it right? Is it not? Are there any questions on that? I saw there were some. Oh, thanks, Russ. Yeah, SVM, support vector machine. And so yeah, very kind of broad overview about how this detector / classifier is working. And we have to make so many different decisions here. So first step, which type of detector do we use? Are we using the blood detector? Are we using something else? And then, you know, when you're doing the blood detector, what window size for your spectrograms? If you're using support vector machine, what probability for each time window should you say, yes, this is a gibbon, no, this isn't a gibbon? Best frequency range. For many target signals, that's actually less challenging to identify. Well, yeah, I'll stick with that, I think. It also depends, like for gibbons, like if you want to try to include some harmonics or not. So anyways, lots of decisions in that case. Another question, which I'm hoping we can talk about this a little bit. How much training data, where should the training data come from? How should you annotate the training data? And so this may be, this may be a little bit more specific to my application in terms of making decisions about annotating training data, but because it has that detector that goes through first and pulls out the signal from the noise, I have been using those signals that I've gone in and manually labeled and using those as training data. But I could imagine doing it in Raven and using Raven selection tables as the training data. So anyways, there's just many decisions in that case. I already mentioned this. So MFCCs have worked really, really well for me. But there are many other types of features that we can feasibly use. Some of the features that Marcelo has developed in his Warbler package would be a good one. There's a lot of other kind of interesting ones out there as well. And then for this specifics, using the machine learning models, so support vector machines are one that I've had the most success with, and I tend to just use the default settings, but that's not a really like systematic, like, well, this is what they said as the default, so this is why I'm using it. But you have to kind of play around with the different parameters of the machine learning models. And I guess one thing that does really influence your subsequent output is that with support vector machines and a lot of these, you can basically have it give you a probability that your observation is in the class gibbon or another class. And so do you want to say, all right, only include signals where the probability is 0.7, 0.8, and then it's still, even though it says it's got that probability, it can still be wrong. So yeah, so anyways, lots of decisions that go into these types of things. But so I have made some decisions, some of them kind of systematic, others just based on the default. And so it presently will give an output of, it's not a selection table, but it's, it's a text file that gives you information on where the detections are. You'll see the probability here is pretty low. So that's something that's a little bit wonky. Okay. And so then the other option is to have it output wave files. So you can listen to them. And then something I have been working on, that I want to share with you and maybe get some input about if this seems like something that you would maybe be interested in using. Okay. Okay, can everybody see my R screen now? I'm not hearing anything, so I'm going to assume that's yes. Yes, okay. And so it's not even like in like a function form yet. It's basically just longhand code that I've been playing around with. But we have basically, if I can show you recent, oh, where is it? Test files. Yeah, so here, if you can see my Finder window, so we have this text file, and then we have these wave files. I'm not sure, could you guys hear the sound? Yeah, so it reads, or it puts them into a folder wherever you would like them to go. And so there are options. You could batch read them all into Raven and do something like that. What I've been playing around with is basically there's a function here that is going to look through. Oh, why is it not working? Oh, the joys of coding live. It's just working. Please ignore all the stuff at the top. There we go. Okay, so it's giving basically an output, and the spectrogram quality is not that great, but I do that just so it can process faster. I can tell that's a gibbon. Maybe I'm not sure if the rest of you can, but so I'm gonna say yes. And so you can input, that's gibbon, that's a gibbon trill. So yes. So I'm just gonna go through this quickly to kind of show the functionality. And I was playing around to try to get it so that it was getting lots of gibbons. And so then it saves basically this table now where it will say, is that actually a target signal or not? So yeah, I would love to hear, for those of you that have done any sort of validation, how that worked, what was challenging, what wasn't. And I know Raven does have, and maybe Ashik or Russ, you could comment on I forgot the name of the functionality, but you can do something similar in Raven, right?
Yeah, Raven has a poorly documented set of tools called selection, review, and annotation, which allows you to see a whole bunch of little postage stamps. It's kind of a grid, kind of like what we're all looking at on Zoom right now, our little pictures of each other. But instead, you've got a bunch of little spectrograms, and you can very rapidly go through and label them as in various ways, especially if it's a simple binary yes, no. labeling. It's one of the less polished features of Raven, and a little tricky, but once you learn how to use it, you can go through data very, very fast.
I see there's a question if there's an option to play it. Not yet, but that's a really great suggestion. So I can see about adding that in. I know, and I'm not sure if anybody has, I've played around with getting R to play sounds and it hasn't really. Kathleen, does it work for you? Yeah.
Yeah, there are ways to do it. So I have, in the R package I wrote, I've gone through a similar sort of protocol that you just showed. And you can definitely get it to play from R. It might depend on your operating system. I have found that it will, the solution I came up with was to just write a temporary file. It'll play that, and then it will like delete it after you. Sometimes you'll run into an issue with like, you have to close the file manually before it will let you proceed in R. So there are ways to do it. The other thing that I ran into in doing that type of like automated, really nice validation that you just showed is it was, depending on the signal I was looking for, it's nice to have the option to sort of put a buffer around the signal itself, depending on what type of signal it is. Because some of the signals I was looking for were like kind of short, and I'd be looking at it like, I don't really know. I can't tell just by looking at it if it's, and I want that like 1 second or 2 second buffer. So that was something that I put in, but it kind of depends on your goals and your processing speed. But anyway, good luck. It looks really great and like super fast the way you just
and the buffer, that's a fantastic idea for sure. So I really, really love that. Yeah.
If I may quickly chime in, Dena. I think to Russ's point that the selection review is actually, even though it's not really very well documented within the Raven system at this point, but it's a very powerful tool for annotating a large number of data in a similar fashion that you showed, but it allows for a lot of different ways of annotating, and it also offers the option that Kathleen just mentioned about buffer, and you can also play it. So if you want to do that step in Raven and then bring the annotated table in R for further analysis, that could be a pretty powerful workflow. And going back to Vijay's point, you can actually annotate a lot of data very quickly.
Okay. Yeah, no, that's fantastic. And I know with all of Marcelo's work, transitioning between Raven selection tables into R, I think it's pretty seamless these days. So yeah, thank you. It's a great suggestion. Does anybody else use any other approaches for validating or checking their annotations? We have the Raven selection review. Kathleen said she's done it a lot with her own package. Yeah. Okay. Wait, Laurel, how about you? How do you do your?
Usually, so I've been working with Sham and his Kugu tool where you get Raven selection tables. So then pulling those back onto the original sound file in Raven and going through mostly in a scrolling fashion, just down the table to accept or reject.
Okay. Yep. Yeah, no, that's just, I think you want to be able to look at them quickly and move on to the next one, right? So
we end up doing a lot of filtering in that process to pull out the time signature of some of the sounds. So there's some additional layers there that are hard to do in just straight selection review format.
Yep. Yeah. Okay. Great. Okay. Oops.
I almost feel, I'm not sure if, I just have one more closing thought. I haven't used the Raven validation form, but I am kind of thinking like the ideal situation for me would be to have like a big, grid postage stamp of all of the things I want to validate. And then some of them are like, it's so clear. I know what it is by looking at it. So I can say yes, no, yes, no, yes, no, go across the whole set of postage stamps and then like leave some of them for that more formal review, the like sort of on the cusp cases. I feel like that would be the workflow that I would want in an ideal world. I have not implemented that, but that's what I think I would want.
It seems, yeah. And I think with that maybe it wasn't that automated detection gibbon paper, but I think there are like Shiny apps that people have developed that I imagine that like kind of more interactive kind of thing. Yeah, I could imagine just like having and like clicking like, you know, just subsequently like keep all of these and then, you know, the next one's like look at them more closely later. So yeah, I think, oh, Russ.
That's exactly what you can do in Raven selection review. And especially if you have access to a fairly large monitor, basically the speed that you can go is based in large part on how big a monitor you have. And if you have even it's not so fast on a laptop, but if you have any kind of external monitor, you can often put up like 100 clips on one screen. And if most of them are true or most of them are false, you can with a few mouse clicks, you can label the minority category. You can say, okay, these 5 out of 100 are true. And with basically a single keystroke, you can say, mark all these as true, mark all the others as false. And you can have multi-way classifications. So you can have true, false, and maybe, or, you know, revisit later
Check
to
later.
address Kathleen's point. So, and then you can, you know, that all of that gets saved to the selection table. So then you can go back later and say, okay, just show me all the things I previously labeled as maybe. So it's very supportive of a pretty fast workflow for that.
Mm-hmm. Okay, great, thank you. Okay, transition back here. And so I think actually a lot of the questions related to the automated detector and classifier we've kind of touched on, but yeah, I think one of my big kind of questions right now is, you know, how do we make decisions about how much training data to use? And I don't know if anybody has any suggestions or thoughts about how they how they can make those decisions. I mean, I know as much as you can possibly do is generally probably better, but we have to find that balance. And does anybody have any suggestions or approaches about how they've dealt with that?
Some of the katydid stuff where we're doing something almost like a rarefaction curve where you train with, you know, 50 samples and look at performance, train with 100, train with 150 and see, you know, are you still gaining? Is it mostly flattened out?
I love that idea. So yeah, basically doing some sort of quantitative thing where you show like, are you getting much in terms of gains with using more training data? I love Russ's answer. Isn't the answer always more? But I prefer Laurel's answer where it's like, use a rarefaction curve, try to figure out where that break point is and then call that good. So no, that's a cool
where
approach.
your breaking point is, which may or may not be the same.
Yeah, it's like it never evens out. It just goes on forever.
But you know, if you get 80% with 100 samples and, you know, with 300 samples, you're still gaining 3% for every 100 samples. And yeah, that's pretty good incentive to keep going.
Yep. Yeah.
0.1% for another 1,000 samples. It's like, well, do I really need it?
Yes. Yeah. Okay. That's great. Thank you. Oh, Ashik says our team record is annotating 240,000 raw selection. So maybe just as many as you can do if you don't stop for 7 days straight. Is that right, Ashik?
Yeah, that would actually be a really neat feature in the package. You know, like a drop X command where it's, you know, drop X percentage of my training data and look at performance and plot as a function of how much went in.
Yes. Yeah. No, I think that's a really cool idea. Okay. Thank you. Those are great. Oops. And so Russ, I think you touched on this a little bit in the context of the BirdNet data. And I have kind of a similar question just because my classifier is not going by time window, it's going by kind of like the output of the blood detector. And so should I just basically label all of the outputs of the blood detector, or should I go and do Raven selections and do labeling that way, do with focal recordings versus PAM recordings? There's a lot of different choices that need to be made here. And yeah, I don't know, does anybody have any thoughts about what or suggestions or things that have worked for them?
I think if I understand what you're asking, Dena, I think that the answer is going to depend a lot on what the recordings are like. If you have targeted recordings, like targeted field recordings with a directional mic, where the target species is the main thing in the recording, and you just need to draw boxes around them in the recording. That's relatively easy, and depending on how dense they are, you could use a bled detector to initially draw the boxes, or just draw the boxes. It gets obviously a lot trickier with omnidirectional mic, autonomous recording type things. So I don't think there's a one size fits. Like most things in bioacoustics, there are probably not one size fits all answers. And so, I think this is where I think people's experience with their own recordings and with the tools available to them, and exchanges like we're having right now with many different people, is really important, because it can give you a sense of what's worth trying and what's not, given the particulars of your situation.
Yes, definitely. No, and I like Laurel's idea of, it's like, well, try it out and see. And I kind of have some PAM data, and then I just kind of added some focal recordings into and so maybe like playing, like taking it, like doing it separately, doing them together, and then seeing how it impacts the output. I think those are really great approaches.
Dena, I have a quick question. This is addressing to Laurel. So Laurel, if you're looking at how much training data to kind of ingest into the system, let's say you have an array, and there are different kind of a difference in the array in terms of background information. Are you training different parts of the array to see the performance dynamics? And to add to that, if there are multi-year data, are you testing that performance in year to year as well? Or is it a one general matrix that you're looking for?
Yeah, in this case, we were taking a random subset of the data to increase the size of the training. But then when we were evaluating performance on the test data, we were evaluating both at the data set wide level and in this case, it was different months. So it wasn't year to year, but it was month to month across wet and dry season performance variation.
Thank you.
Yeah. Thanks for that point in those questions. And yeah, it's so interesting, right? We have to make so many decisions here about, do we look at, differences in the space of the recorders or the time? And I mean, ideally we'll look at everything, but I don't know, makes it challenging. Okay. I see Wendy has a question. Yeah. So when training, doing annotations for your training, do you focus on kind of binary, gibbon, not gibbon, or do we try to capture more information, like including the SNR of the signals, or, if they're overlapping, or if there's other background noise?
Yeah, I'm just wondering. And also Vijay had his hand up. So I'm sorry I rushed ahead of you, Vijay, and hopefully you get to
sorry. I saw
No,
his
it's
hand
okay. It's hard to see everything in the Zoom world.
Yeah.
But yeah, I was just kind of wondering, I'm sort of like an over annotator by nature. And so if I'm going through and making these selections, I just want to capture as much information as I can. So thinking about having some kind of coding approach, whether it's additional annotation, or what do we call those things? I'm blanking on the terminology here, but additional columns for annotations where you might have overlapping other gibbon or overlapping not a gibbon, or this is a really faint call, this is a really good call, or do people use something like one of the SNR measurements that's available in Raven to, sort of be a way to filter out things that have like a low signal to noise ratio when you're training models and evaluating different models. So those are always the kinds of things that that vex me making those decisions. So I was wondering if anybody had experience or advice on that front.
I think in some ways it depends how readily available your annotation sounds are. So if you have lots and lots of them, like with the insects, we had thousands to work with. And so we tended to go for speed over detail and just, take really clean ones and move as quickly as we could to build up. Doubling the sample size did more than having really detailed information on the individual annotations. But, if you only have 50 annotations to work from and it's really hard to find them, it might make sense to keep notes on the quality of the individual annotations so that if you aren't getting the performance that you want, it's really easy to go back and exclude the 20% that are the lowest quality, or to add them back in for sample size and to try to refine the data set. That's just my personal sense. Other people may have experience there also.
One comment I'd add to in response to Wendy is just to keep in mind, if you are going for more detailed annotations than target yes, target no type things, than binary or even a multi-way classification. If you want to have other annotations that indicate something about signal quality, presence or absence of overlapping signals, and so on and so forth, remember that it will be, if you want to do any kind of large-scale automated processing of your annotation data later, it will be much, much easier if the contents of every column is just sort of basically like a multiple choice value. I mean, the last thing you want to do is, like, for automated processing, is type free-form comments into an annotation field. You can do that. And if a human is looking at it, that might be useful. But for processing large amounts of data, you really want your annotations to be, basically each column, the values that go into each column should be drawn from a set of predefined values, if that makes sense. And that will just make things downstream processing much easier.
Absolutely. Thank you both.
I also used thing before, and we had two sort of classifications. So you could do a rating from like 1 to 5 or 1 to 10. If you wanted, it was optional. And then you could also classify it based on what it was. And we were finding multiple species in the same, or species and call types in the same recordings. And so you could either label it like A or B, like simplistic. And then each of those corresponded to whatever, if it was a different call type or a different species. But the labeling of whether it was like a high quality or low quality helped if you were ever going to go back and want either exemplars for training data sets or exemplars for, like a presentation or whatever.
So, Dena, I had a quick question on the training data itself, and thinking about the other end of the spectrum, which is in some cases, even after recording for like a year or so, you're not going to get enough training data for some rare species. So is there any option to add any sort of data augmentation features which might help improve the accuracy? I know people have been doing that for some neural networks now, but I'm not sure what's the way around, especially really rare bird calls like great hornbills or something like that.
I guess I can't really comment on the data augmentation, but I could imagine, I mean, it comes also down to like how variable the calls are. But if you use something like a template detector, you only need one call or a few calls. So I guess that could also influence the type of detector and classifier that you're using if you don't have a lot of training data. And I'm not sure, does anybody have experience with the data augmentation with the neural nets or anything? Some of our other team members might. So if you want, you can send an email and I can put you in touch with them. They're more of the deep learning guys than, than I am. So. Okay, so I think we're getting close. Oh, wow, we're almost at time. Thank you all for this great discussion. And then I guess, yeah, we have talked about validation a little bit. So I'll just finish really quickly to show you the next package that's very much in progress. So I don't really need much time to discuss this, but so there's two main functionalities. And after watching Marcelo's talk the other week, I realized that this function to calculate MFCCs already exists in one of his packages, but I will probably keep it just because it's formatted in a way that's easy for me. And then the other one is biplot visualization, and you can do it with or without spectrograms. Again, there's a lot of different decisions that can be made. So what type of algorithm are you going to use to create the biplot? Right now it uses UMAP. That's just one that I've been using for a while, and I'm pretty happy with how well it works for data visualization. And then the labels can be colored by the points can be colored by labels, so gibbon individual or call type, for example, or you can use an unsupervised clustering algorithm to have it assign cluster ID and then color it that way. And then, so this is just an example one here I did with female gibbon calls, and each of the colors around the boxes of the spectrograms represents the female ID. So that can be a good way to look for patterns of individuality, etc. Another, and this was just a toy example, another kind of fun approach or application I could imagine for this is to read in all of your different sound types and then use some sort of unsupervised clustering thing like this to see, like, in your soundscape, how many different sound classes are there. So yeah, with that, I think we're right about at time. If anybody has any last things they'd like to add. And I appreciate everybody's input and discussions on this. And yeah, it's been a huge learning process for me. So it's nice to have people like Kathleen who have already published our packages that can give, it's one thing to like write up the code. It's another thing to like put it into a package that is easily used for other people. So yeah, it's definitely been a learning process for me, but okay. And then Laurel, who do we have in 2 weeks from now?
So on deck, we have Juan Sebastian Oya, who will present scikit-mad, which is a Python toolbox for soundscape analysis. So more acoustic tools coming up. And yeah, we are currently filling the schedule for the coming sessions. Beyond that, Shyam has agreed to do a session on his Kugu tool for training machine learning models from your data on Google Drive, but we're taking suggestions and ideas. A few people have sent names of either packages or presenters that they would love to hear from, but this is also your chance to nominate somebody, even if you don't know them, and it's just like, oh, this is a tool that I might want to use. We're happy to reach out and see if they're willing to hop on and chat with us. So thank you all for coming out. Thank you, Dena, for the cool talk. And yeah, it was a fun discussion today. It's nice to have the back and forth and to see everybody. So I hope you all have a good week.
Thanks everybody for contributing. I really appreciate that. So we'll see you all next time.
Soon. Take care.
Thank you.